Today we're going to explore epistasis in the Primula plant. We'll start with some basic definitions. Simple Mendelian inheritance should be the form of inheritance that everyone is most familiar with. Epistasis, however, is a different type of inheritance that results from the masking of a gene locus. When phenotypic expression is altered from an epistatic event, we call it a hypostatic gene. There are many types of epistasis, as shown below. We're going to focus on single dominant epistasis. The Primula flower, known as a primrose, is a popular flower that is found throughout the northern hemisphere. The plant is most well known for its wide variety of petal colors. However, scientists do not know what causes the phenotypic variation. Researchers Yamamoto, Honda, Ihara, and Setaguchi tested 60 microsatellite markers for polymorphism. However, the markers that they tested did not show a strong correlation with epistasis, and they determined that genetic drift and bottleneck events must have been the cause of the color variation. Other researchers, such as Shipnuov, Kosenko, and Volkova, tested various populations of the plant for morphologic and geographic determination of colors. They were unable to conclude if morphological characteristics such as the calyx, corolla tube, and petal limb length correlated with color, though they did find geographic trends in their data. These trends show that the percent of magenta flowers increased and the percent of light flowers decreased as their measured distance increased. An old study by Harbord and Sherritt suggested that anthocyanin concentration was responsible for petal phenotype. They found that methylation of the anthocyanins could be masked by other copigmentation factors such as peonidin and pelargonidin. Based on this study, the K gene was found to affect the concentration of methylated anthocyanins. While these results are not conclusive, it shows that the Primula petals should be investigated further.
Primula examples. For the following examples, we'll only be looking at the K and DZ alleles. We'll also be simplifying DZ into just D. The big K allele codes for high concentrations of anthocyanins and melvidin, which makes flowers appear red. It's dominant over the little k allele, which codes for much lower concentrations of pigment and makes them appear white. The big D allele mainly codes for pelargonidin, a pigment that produces an orange color. It also codes for higher concentrations of anthocyanin. The little d allele codes for the orange pigment in lower concentrations. These are the possible genotypes. So, big K results in scarlet flowers due to the high anthocyanin concentration. Little k and big D code for orange flowers because the orange pigment from big D has dominance over the white flowers from little k. Big K and little d code for scarlet flowers because the red pigment in the big K allele has dominance over the little d. Finally, little k and little d code for weaker anthocyanin and pelargonidin concentration, making the flowers appear coral. So, what mode of epistasis is occurring? We can determine this by looking at a dihybrid cross between two plants that are heterozygous for both alleles. Here is the Punnett square for a dihybrid cross between two plants that are heterozygous at both alleles. We know that big K and big D code for scarlet flowers. Big K and little d also code for scarlet flowers. Then, little k and big D result in orange flowers. And lastly, little k and little d code for scarlet flowers. This gives us a ratio of 12 scarlet flowers, 3 orange flowers, and 1 coral flower. This indicates that the mode of epistasis is dominant suppression, or single dominant. Let's try an example. What is the phenotypic ratio expected in the following dihybrid cross? Here's the expected Punnett square. Big K and big D and big K and little d will be scarlet. Little k and big D will be orange. Little k and little d will be coral. This gives us a ratio of 8 scarlet flowers, 4 orange flowers, and 4 coral flowers. Thank you for watching!